So today we will discuss about the MSC complex or the major histocompatibility complex, and then we will see the how the antigen processing and presentation happen, okay, through the MSC complex. So in every mammalian species, the data means to date possesses a tightly linked cluster of genes, that is the MSC complex, whose product play a role in the intracellular recognition and the discrimination between the self and non-self. Okay, so uh, suppose one antigen comes inside our body. Okay, so there is a mechanism which is happen through the major histocompatibility complex, and suppose this is the antigen and this is your cell. Okay, so inside the cell, how like our body recognize that this particular antigen belongs to our body or it comes from the outside. Okay, so this MSC molecule play important role to recognize those antigen, to recognize or to helping the T cell to recognize this antigen as a self or non-self. Okay, because this MSC will presented this antigen to the one of the T cell, either it will be the T helper cell or T cytotoxic T cell. Okay, so this T cell will know that this is self antigen or the non-self antigen through the help of MSC molecule. Okay, if it is like the MSC participate in the development of humoral as well as the cell mediated immune responses. Okay, while the antibodies may react with antigens alone, but the T cell only will recognize this antigen when it is bound with the major histocompatibility molecule. Okay, so this MSC molecule that is that function in this recognition event, which is termed as the antigen presentation, are highly polymorphic in nature. Okay, and it is a glycoprotein which is present on the cell membrane. Okay, and this MSC complex bind with the peptide derived from the pathogen and display them to the cell surface, which is recognized by the appropriate T cell. Okay, as I told you that this MSC molecule will firstly process this antigen and presented this antigen to the one of the T cell, either it will be the T helper cell or the cytotoxic T cell. Okay, antibody mediated response doesn't need this MSC molecule. Okay, because it comes later. Okay, means firstly the T cell mediated immune response comes in the in the scene, but after that, if it is not means tolerated by the T cell immune response, then antibody mediated immune response will play role. Okay, so for that, like firstly our innate immune system will work, then after that our adaptive immune system will work. Okay, in adaptive immune system there is two type of response. One is the antibody mediated response. Okay, and another one is the T cell mediated response. So this will come later. Firstly T cell mediated response will work, and once it is not tolerable by this T cell, then antibody mediated response will work. Okay, so this T cell will only get to know that this antigen is belongs to our cell body system or it comes from the outside through the help of MSC molecule. If this antigen is bind with the MSC molecule, then it can called as a non-self antigen. Okay, and if it is not means suppose in inside our body. Okay, so in our body antigen never will bind with the MSC molecule. Okay, only the autoimmune condition the self antigen will bind to the MSC molecule and it will be presented to the T cell. Otherwise the non self antigen mostly bind to the MSC molecule and it is presented to the T cell. Okay, and these MSC molecule are highly polymorphic in nature because they has to means bind with different kind of antigen, so that's why their nature is highly polymorphic, means it is highly diverse in nature. Okay, and it is glycoprotein. Okay, which is present on the most most of the immune cell, I can say. Okay, and most of the nucleated cell. So there are three major class of MSC molecule: class 1 MSC molecule, class 2 MSC molecule and class 3 MSC molecule. Okay, so this MSC class 1 MSC molecule is presented all nucleated cell which is present inside our body. Okay, like it can be immune cell as well as the non-immune cell also it will be presented, and it means all the nucleated cell in RBC is not having the nucleus, so this MSC1 will be not present there, but apart from there it is present on all type of cell present inside our body. Okay, and the major function of MSC1 genes is it is presenting the peptide antigen to the cytotoxic T cell. Okay, this you has to remember like two things. Firstly, it is present in the nucleated cell, all the nucleated cell the MSC1 gene will be always present. Okay, the second thing is the it will present the antigen to the cytotoxic T cell, or we can say that CD8 T cell. Okay, it present peptide to the CD8 T cell or cytotoxic T cell. Okay, whereas this class 2 MSC genes it is present only on the antigen presenting cell. Okay, what are the antigen presenting cell? The macrophages, dendritic cell, and B cell. And what type of cell is called as the antigen presenting cell, which who generally do the phagocytosis mechanism. Okay, that type of cell is called as the antigen presenting cell, and these cells are macrophages, dendritic cell, and B cell. Okay, that you have to remember. Okay, and this MSC2 molecule means present only on the antigen presenting cells, and it present the peptide to helper T cell. Okay, helper T cell it present the peptide, and it is also called as the CD4 T cell. Okay, CD4 T cells. This is called as the CD8 T cell. Cytotoxic T cell is called as the CD8 T cell, and this helper T cell is called as the CD4 T cell. Okay, these are the surface marker of the T cell. Okay, then third class is which is not very much having the biological means not having in the role of antigen presentation and yeah, these things, but it encodes certain kind of secreted protein or certain kind of secreted cytokines which is having some immune function. Okay, so here in the organization of MSC, if you will see, this is the mouse MSC complex. Okay, so this is the MSC1, MSC2, this is the MSC3. Okay, so the locus of MSC1 is called as the like here you can see that is the D locus, and 3 is located on the S locus, 2 is located on the IA or IE locus. Okay, and this one is also located as the K. So these these 3 MSC genes is highly polymorphic, and it contain almost 240 genes inside their locus. There is 240 gene which is consist in a MSC molecule. Okay, so like in case of class 3, it is coded the TNF alpha and TNF beta type of cytokine, and apart from that it can also code the complement system proteins also. Okay, and these two is majorly the class 1 and class 2 is majorly play role in the antigen presentation. Okay, and this class 3 is used for the secretion of pro-inflammatory cytokine like the TNF alpha and beta and the complement system related proteins. Okay, so these are the 3 major class. Class 1 generally present the peptide to the CD8 T cell or cytotoxic T cell, and class 2 MSC antigen will show the peptide to the CD4 T cell or helper T cell. Okay, and this is present only on the antigen presenting cell, whereas this MSC1 gene will be presented in all type of nucleated cell. Okay, so this is the structure of MSC1 and MSC2 molecule. Okay, so in case of MSC1 molecule, which is consist of 2 polypeptide chain, a larger, there is two polypeptide chain. Okay, so the domain is alpha 1, alpha 2, alpha 3 and beta 2 microglobulin. Okay, so these three will consider as a single polypeptide chain, and this will be considered as a another polypeptide chain. Okay, so this is the two polypeptide chain. Okay, and it encoded on the chromosome number 6 of the human. Okay, and this smaller chain is called as the beta 2 microglobulin, which is encoded by the chromosome number 15. Okay, and this class 1 chain consists of a single polypeptide composed of three extracellular domain named as the alpha 1, alpha 2 and alpha 3. Okay, so these are the three, in the single polypeptide chain there is three chain. One is the alpha 1, alpha 2 and alpha 3. All the three chains are outside of the cell. Okay, or we can say it is the extracellular domains. Okay, and there is the transmembrane region that anchor in the plasma membrane. Okay, so there is the transmembrane region which is spanning towards the inside of the cell, and there is a short intracytoplasmic tail. Okay, so this is the short cytoplasmic tail, this is. Okay, and there is one beta 2 microglobulin. Okay, this is non polymorphic in nature. Okay, and it is non covalently bound to the alpha chain. Okay, these three are the alpha chain. So this beta 2 microglobulin is bind to the alpha chain through the non covalent interactions. Okay, and yeah, this is the major structure, and here you can see that the alpha 1 and alpha 2 is having the antigen binding site. Okay, this is the peptide binding cleft which is made by the alpha 1 and alpha 2 in case of the MSC1 molecule, that you have to remember. Okay, this alpha 1 and alpha 2 is antigen binding site or antigen binding domain in case of the MSC1 molecule. Okay, and this region is highly polymorphic in case of the MSC1 molecule, alpha 2 and alpha 1. Okay, why it is highly polymorphic? Because it is the only region which are going to interact with the different kind of antigen. Okay, so that's why this alpha 1 and alpha 2 is highly polymorphic in nature in case of the MSC1 molecule, and it make the peptide binding cleft. Okay, and it has two polypeptide chain. One is encoded by alpha 1, alpha 2, alpha 3, and another one is the beta 2 microglobulin. Okay, these three comes under one polypeptide, one peptide chain, and this comes under the another peptide chain. And yeah, so these two polypeptide chain link together via the non-covalent interaction. Okay, and in case of the MSC2 molecule, there is also two polypeptide chain. Okay, and which is encoded by chromosome number 6. Okay, and the A and B chain each consist of two extracellular domain referred as the alpha 1 and alpha 2 and beta 1 and beta 2. Okay, here the alpha 1 and alpha 2 domain and beta 1 and beta 2 domain. Okay, so this is also having the transmembrane region and one cytoplasmic tail inside of the cell, and the extra here the alpha 1 and beta 1 is making the peptide binding cleft, and it is the highly polymorphic in nature, alpha 1 and beta 1 in case of the MSC2 molecule. Okay, don't confuse, like in case of MSC1 it is alpha 1 and alpha 2, in case of the MSC2 it is alpha 1 and beta 1. Okay, you have to remember this thing. Okay, these two domain is highly polymorphic in nature, and it make the peptide binding cleft. And here in case of the MSC1 molecule, alpha 1 and alpha 2 make the peptide binding cleft, and it is the highly polymorphic in nature. Okay, and another dissimilarity is that in case of MSC1 there is beta 2 microglobulin, whereas means here the beta 2 microglobulin is very short in size, right? But in case of the this MSC2 molecule, this beta 2 it is not microglobulin. Okay, it is the same as the remaining three. Okay, by size wise. Okay, so there is two polypeptide chain, alpha 1 beta 1, alpha 2 beta 2. But in case of the MSC1 molecule, there is alpha 1, alpha 2, alpha 3 will make one chain, and beta 2 will be the separate chain. Okay, and this MSC1 molecule present the peptide to CD4, CD8, and this MSC2 molecule will present the peptide to CD4. Okay, so there is the difference between the MSC1 and MSC2. So this the polypeptide chain, a single chain in case of the MSC1, it is around 44 to 47 kilodalton, which is non-covalently linked to the beta 2 microglobulin chain, which is 12 kDa. Okay, and in case of MSC2, a single chain, a chain, okay, A chain or alpha chain is around 32 34 kDa, and it is non-covalently linked to the single B chain, which is around 29 to 30 kDa. Means these two chains are almost equal in size. But in case of the MSC1, the alpha chains are means bigger in size in comparative to the beta 2 microglobulin chain. Okay, and the distribution in case of the MSC1 molecule, it is present in all the nucleated cell, and it is only present on the antigen presenting cell. Okay, and composition of antigen binding cleft, we have seen that alpha 1 and alpha, sorry, here wrongly written. Okay, this one, alpha 1 and alpha 2 domain make the antigen binding cleft, or antigen binding site, or in case of the MSC2, it is alpha 1 and beta 1 domain. These domains are highly polymorphic in nature, because it is going to interact with the different kind of antigen. Okay, and then it binding site for T cell co-receptor, it will, MSC1 will bind to the CD8 T cell. In case of CD8 T cell, it will bind to the like in case of the CD8 bind, this T cell will bind to the alpha 3 region of the MSC1. Okay, that also a very important question, like through which domain it will interact with the CD8. So it is the alpha 3 region. Okay, there is three domain, right? Alpha 1, alpha 2, and alpha 3 in case of MSC1. Okay, so the alpha 3 will participate in the T cell recognition. Okay, whereas in case of the MSC2, it is the beta 2 region. Okay, that you has to also remember, like the T helper cell will interact with the beta 2 region of the MSC2. Okay, then in case of the MSC1, the peptide size will be around 8 to 9 residues. Okay, and the standard one will be 9 amino acid, and in case of the MSC2, it is around 13 to 25 amino acid. Okay, the peptide length. Okay, and the here the standard size is 15 amino acid. Okay, and in case of how we nomenclature this MSC1, we generally consider it as a HLA molecule. Okay, HLA-A, HLA-B, HLA-C, and in case of the MSC2, it is HLA-DR, HLA-DQ, and here DP. Okay, so this ABC, DR, DQ, DP, these are referring to the locus where the MSC molecule is present on the chromosome. That locus is called as the ABC either, or if it is MSC2 molecule, then it is called as the DR, DQ, DP. Okay. So this is the difference between the MSC1 and MSC2 molecule. Then what is the antigen processing? So proteolytic cleavage of protein by enzymes, that is called as the proteases, okay, into a small fragment, or we can say this is the antigen peptide and their association with MSC molecule by the antigen presenting cell. This is an active process requiring energy. Okay, so this antigen processing is what? When this antigen is chopped into a small fragment and make it enables to for the presentation. Okay, so that is that process is called as the antigen processing. And what is antigen presentation? The presentation of this processed antigen on the surface of either APC or all the nucleated cell to the any kind of T cell. That procedure is called as the antigen presentation. Okay, So there is two type of antigen processing pathway. One is the exogenous antigen processing, and another one is the endogenous antigen processing. Okay, so what is the endogenous antigen processing? So endogenous proteins are processed in the cytosol or in secretory vesicle and presented on the class 1 MSC molecule to the CD8 T cell. Okay, so these endogenous proteins are present inside of our body, suppose like bacteria, virus. Okay, so when they enter into our body, their protein will be processed inside our body. Okay, so that type of antigen is called as the endogenous protein, and which is processed through the MSC1 molecule to presented to the CD8 T cell. Okay, whereas this exogenous protein are the processed in endosomes, and exogenous means which is comes from the outside. Okay, means the processing itself is happen in the outside. Okay, and it is presented by the class 2 MSC molecule to the CD4 T cell. Okay, then endogenous antigen are derived from the proteins processed inside the cell. These include altered cell protein antigen, like sometimes it could be the self antigen also, like the tumor antigens, and non-self proteins antigen, like the viral antigens. Okay, viral or bacterial antigens or parasitic antigen. Okay, then this endogenous antigens associated with the class 1 MSC molecule that activated the cytotoxic T cell or CD8 T cells for killing infected cell and tumor cells. Okay, and these endogenous antigens can be processed and presented by any nucleated cell. Okay, as we earlier discussed, it can be means presented by the any kind of nucleated cell, because MSC1 molecule found in all the nucleated cell. Then there is exogenous antigen. Exogenous antigen are derived from the protein produced outside the cell. Okay, so outside the cell whatever antigen is processed, that is called as the exogenous antigen. These include various bacterial, viral, protozoan, fungal and parasitic antigen which are derived from the outside body. Okay, some bacteria and some virus which is processed outside our means which is not host dependent, that type of organism processed outside of the body. Okay, so these antigen are associated with the class 2 MSC molecule, and it activate the helper T cell, or it is the CD4 T cell. Okay, so it will presented to the helper T cell or CD4 positive T cell, and this type of antigen is presented by the APCs or antigen presenting cells. Okay, so then we will see like how the endogenous antigen processing pathway follows. Okay, and this endogenous antigen processing is also called as the cytosolic pathway. Okay, there is three major steps. First one is the processing of antigen into peptides, then assembly of MSC and peptide loading complex, then third is the peptide loading and MSC peptide transport to the plasma membrane. Okay, so here you can see that this is the protein. Okay, and it is the inside the cell. Okay, here you can see it is the cytosol, so it is the inside the cell. When protein comes inside the cell, the protein is labeled through the ubiquitin protein. Okay, when this protein is tagged with the ubiquitin protein, this ubiquitin will lead to the this protein to proteasomal there is a proteasomal type of what I can say, this is the kind of uh, protein, big assembly of the proteins which is called as the proteasomal degraded machinery. Okay, so when labeled the ubiquitin molecule here, it will work as a signal molecule to lead to this peptide to here. Okay, this proteasomal degradation machinery. Okay, so after means process through this protein in from the inside of this proteasomal degrading machinery, it will form this type of peptide which is around 8 to 11 amino acid in length. Okay, and after the peptide is processed into small pieces, then this through energy gradient, it will lead to the means it will moves from here cytosol to this endoplasmic reticulum. through the help of this tap molecule. Okay, this tap molecule is transmembrane associating proteins, which will create help in the creating pores to enter this peptide inside the endoplasmic reticulum. Okay, so once this peptide will comes inside the endoplasmic reticulum, there is already the MSC1 molecule is present here. Okay, this green one. Okay, MSC1 molecule is already present with it is stayed there with the help of chaperones, that is the calreticulin, calnexin, and one more thing is tepacine. Okay, and this is the complete mechanism how it works. Like initially the class 1 molecule is bind with the calnexin. Okay, and it will create the calnexin associated class 1 MSC alpha chain. Okay, then beta 2 microglobulin will attach here. Okay, and form the calreticulin tepacine associated class 1 MSC molecule. Okay, so once the peptide will come inside the endoplasmic reticulum, this chaperone will remove from the MSC molecule, and it will bind to the class 1 MSC molecule. The peptide will bind to the MSC1 molecule, and this chaperones will sequentially removed. Okay, from the class 1 MSC molecule. Then once the class 1 MSC molecule bind with the peptide molecule, it will transport to the through vesicles, it will transport to the endoplasmic reticulum to Golgi body, then Golgi body to plasma membrane. Okay, so in the plasma membrane, this antigen will be presented to the CD8 T cell. Okay, understood? There is 5 steps. First is the ubiquitination of this protein. Okay, and for ubiquitination means the ubiquitin tag will add here, and this will work as a signal for the protein to move to the proteasomal mediated machinery. Okay, so inside of the proteasomal, when this protein will pass through this machinery, it will chopped into small pieces of the peptide. Okay, which is around 8 to 11 amino acid in length. Okay, so if then there is in the endoplasmic reticulum, there is a tap molecule that is helping in the creating pores to move the peptide from outside of the endoplasmic reticulum to inside of the endoplasmic reticulum. Okay, so it is also work as a regulatory protein, because if the peptide length is not 8 to 11 amino acid length, then it won't allow this peptide to go inside. Okay, it will inhibit there itself. Okay, so for going inside the endoplasmic reticulum, it should be around 8 to 11 amino acid in length. Okay, and after coming inside of the endoplasmic reticulum, there is MSC molecule which is attached to the different type of chaperones, that is called as the tepacine, calreticulin, and calnexin. Okay, so this once this peptide will bind to the MSC molecule, this all the chaperones will remove from or detach from the MSC1 molecule. Okay, and after binding of the MSC1 molecule, this complete complex will be packed into a vesicle, and it will be moved to the endoplasmic reticulum to Golgi body, and Golgi body to plasma membrane of the cell. Okay, in the plasma membrane, it will be presented to the CD8 T cell. Okay, so this is the cytosolic pathway, or we can say that endogenous antigen presentation. Okay, there is exogenous pathway, which is also called the endosome pathway. Okay, don't confuse with the word here. The endogenous antigen is presented by the cytosolic pathway, but here the exogenous antigen is presented by the endocytic mediated pathway. Okay, because here here endosome will form, so that's why this pathway name is called as the endocytic mediated pathway. Okay, so there is also 3 major steps. First one is the uptake and processing of exogenous antigen, then MSC assembly and transport to the peptide loading compartment, and third one is the peptide loading and MSC peptide transport. Okay, so transport to the plasma membrane. So like inside of the like suppose there is antigen which is already processed, it will be through the clathrin coated vesicle, it will be means internalized, okay, inside the cell, okay. And it will create the vesicle for the internalization of this antigen, that is clathrin coated vesicles, okay. This clathrin coated vesicle will form the early endosome, okay. And then middle endosome, and there is the late endosome, okay. And meanwhile in the endoplasmic reticulum there is the class MSC class 2 molecule, okay. MSC class 2 molecule is present, which is already bound with the invariant chain. This yellow color which you are seeing, that is called as the invariant chain here, okay. So this is the MSC2 molecule, and this is the invariant chain which is already present as a bound form inside the endoplasmic reticulum, okay. So when the antigen is internalized and it forms the late endosome, then this MSC molecule should be fused with this late endosome, okay. So for that, when it fused with the late endosome, what will happen is, firstly it will, late endosome, what will happen is, in the late endosome, the invariant chain will be dissolved, okay. This tail part is dissolved because of the high pH, that is the high acidic pH, that is the 4 to 5 pH, okay. In that case the invariant chain will be dissolved, and only a small part of this invariant chain is still attached with the MSC2 molecule, which is called as the clip molecule, okay. And after in this form, what will happen is, this late endosome of the this late endosome of the antigen will fused with the this late endosome of the MSC2 molecule, okay. And in when these two endosome will fuse together, then the clip molecule will remove from here, and the antigen will go and bind to the antigen binding cleft of the MSC2 molecule, and then through the vesicle process it will reach to the plasma membrane of the cell and presented to the CD4 T cell, okay. Understood? In case of the like MSC1 molecule, the process is similar, but here like the MSC is attached with different kind of the chaperones which help to maintain this MSC molecule to like not bind with any other kind of the peptide, and it provide the stability to stay here, okay. So once the molecule will come and bind to here, it will be transported to through vesicle to the plasma membrane, in case of the MSC1 molecule. In case of the MSC2 molecule also, the like the presentation is same almost, but there is the other type of protein play role. Like in case of the MSC2, there is invariant chain which already block the block the antigen binding cleft, because so that it can't be like interact with other type of antigen, okay, self antigen or other type of antigen, okay. So once this antigen comes inside our body and create the late endosome, then only it this MSC2 molecule will get signal and go to the late endosome phase, where the pH is very high, acidic pH. In this high pH, this invariant chain will be digested, and only a small part of the invariant chain will be still attached to the MSC2 molecule. And when these two vesicle will fuse together, or two endosome will fuse together, then only this invariant chain clip molecule will remove out, and the antigen will bind to the MSC2 molecule, okay. And in this condition, it will be through the vesicle form, it will be reached to the plasma membrane of the cell, and here it will presented this antigen to the CD4 T cell, okay. T cell. So you have to just remember these two pathways, like how means this exogenous pathway is happen, and how the endogenous antigen pathway is happen, okay. So this whole procedure you have to remember like in CSIR exam point of view, or in get question in 3 numbers question, it can be asked, like in a statement wise question can be asked, what is the correct sequence of the exogenous pathway or the endogenous pathway, like that. Okay, so just remember this pathway. So this is the complete procedure of the endogenous pathway and exogenous pathway. Here you can see the endogenous antigen which is present inside of the cell go through the proteasomal degradation, and it will create 8 to 11 amino acid length peptide, and through tap molecule it will enter into the endoplasmic reticulum, where the MSC1 molecule attached to different type of chaperones, and when it bind to the antigen, all the chaperones will remove out, and this through the vesicle, this antigen and MSC complex will go to the Golgi complex, and then it will go to the cytosol, and through cytosol it will presented on the cell surface or the plasma membrane of the cell to the CD8 T cell. Okay, in case of the exogenous pathway, the antigen is already like processed outside of the body, and through clathrin mediated vesicle endosome it will comes inside of the cell. Okay, where the MSC2 molecule in the endoplasmic reticulum already is attached with the invariant chain, which is act as the inhibitor or blocker of the any kind of peptide binding. Okay, so this will then go to make the late endosome, where the pH is acid pH is high, and in this acid pH this invariant chain will be digested, and only the small part of the invariant chain, which is called as the clip, okay, it will remain here. And once these two endosome will fuse together, this clip molecule will also remove, and it will bind to the MSC2 molecule and presented to the presented to the CD4 T cell. Okay, so this is the complete procedure. Endogenous antigen are presented on the cell surface along with MSC2 molecule, MSC1 molecule, FC receptor, complement receptor. What will be the correct answer? Yes, the correct answer will be MSC1. Okay, it is processed inside of the cell. It is called as the endogenous antigen, which is presented by the MSC1 molecule to CD8 T cell. Whereas in case of the MSC2, it is presented the exogenous antigen to the CD4 T cell. Okay, whereas these two receptor are and this receptor is present on the antibody. Okay, FC domain we have seen in previous classes, we have seen the receptor is present on the antibody, and this complement receptor is play a role in the complement system. Okay, so the correct answer of this question will be MSC1.